Approximately 70 to 100,000 Americans have sickle cell disease. This is an inherited blood disorder caused by a mutation in the hemoglobin protein that causes red blood cells to form a sickled crescent shape. These cells can break apart, clump together, and cause blood clots, contributing to anemia, severe pain, and risk of stroke. There are 120 million people affected by sickle cell disease worldwide. In Africa, half of the 300,000 children born with sickle cell disease each year die before the age of 5. In December 2023, the United States Food and Drug Administration approved 2 types of gene therapy to treat sickle cell disease. The treatment modifies the patient's cells so that they produce a healthy form of hemoglobin, and for many patients, this could be a cure for this painful and deadly disease. However, the estimated cost for these treatments for one patient is $2.2 million for one of the treatments and $3.1 million for the other. This raises serious questions about access to this life-changing treatment. In this video, we will examine what is sickle cell disease? How does gene therapy work? How is sickle cell disease treated with gene therapy? And finally, how do insurance companies determine what to cover? Stick around to the end of this video where we consider the question, should insurance companies pay for gene therapy to treat sickle cell disease? In healthy individuals, our red blood cells appear like donuts in which the hole is not removed, and these are packed with the protein hemoglobin. This hemoglobin protein carries oxygen from our lungs to the cells that need it. The red blood cells are very flexible and easily move through our blood capillaries. In individuals with sickle cell disease, there's a mutation in the hemoglobin gene, resulting in a single amino acid change out of the 146 amino acids. This minor change in the protein structure of hemoglobin causes red blood cells to take on a sickle shape and be much less flexible. These altered red blood cells can clump, can block blood flow, contributing to extreme pain, organ damage, and increased risk of strokes or heart attacks. The proteins in our body are built using instructions from our DNA. When there is a mutation or change to the DNA, these instructions may be altered, resulting in a slightly different protein that may not function properly, as is the case with sickle cell disease. Healthy red blood cells contain an adult form of hemoglobin known as hemoglobin A. This hemoglobin allows the red blood cells to remain round and flexible to squeeze through our smallest capillaries. A person with sickle cell disease produces a different form of hemoglobin known as hemoglobin S. This hemoglobin forms rigid clumps, causing the red blood cells to have a sickled C shape, which will cause them to clump together within our bloodstream and potentially clot and interfere with the normal blood flow. Red blood cells are continually formed from special blood stem cells located in our bone marrow. As the cell develops, they accumulate the hemoglobin protein and move into blood vessels. The red blood cells will continue to circulate through our bodies until they become old or damaged, at which point they are destroyed. Healthy red blood cells can live for around 120 days, but the red blood cells in a patient with sickle cell disease may only last for 10 to 20 days. So the loss of these red blood cells can contribute to anemia. One way in which sickle cell disease is treated is through blood transfusions, in which healthy blood cells from a donor are matched to the recipient and help replace the damaged or lost sickled red blood cells. These blood transfusions are only a temporary replacement, as the donated blood cells will also need to be replaced as they get old or damaged. A permanent cure for sickle cell disease is possible and involves a matched bone marrow transplant. These patients will undergo chemotherapy to kill their blood-forming cells and then receive blood-forming stem cells from a donor. This procedure requires a match between the donor and recipient to prevent the body from rejecting the transplanted cells. The sickle cell treatments approved by the Food and Drug Administration in December 2023 involve using gene therapy to edit the DNA of a patient's cells. In gene therapy, the DNA molecule is repaired so that an edited gene produces the correct protein. For sickle cell patients, the gene-edited cells will produce a working hemoglobin protein. One of the gene therapy treatments uses an exciting set of molecular tools known as Crispr. This system allows for easily and precisely targeting and correcting specific sequences in the DNA molecule. Emmanuel Charpentier and Jennifer Doudna received the Nobel Prize in Chemistry in 2020 for their pioneering work on CRISPR. This was the first time that two women shared the prize in chemistry. It is important to recognize that the type of gene therapy approved for treating sickle cell disease is an example of somatic gene therapy. This means that the DNA is edited in a specific set of body cells, in this case, blood-producing cells. This is different from germline gene therapy, where the DNA could be changed in the embryo. This would result in DNA changes that would be carried in a person's eggs or sperm cells and be inherited. This type of gene therapy is used in creating animal models for research, but is not used in humans because of the concerns of changing the DNA and traits of future generations. As we look at the use of gene therapy for the treatment of sickle cell disease, we see that this is a prime example of personalized medicine in which the process alters the DNA of a patient's cells. Physicians collect blood-forming cells from the patient's bone marrow. The DNA of these cells is then edited in the laboratory before being returned to the patient at a later time. While the patient's cells are being treated in the laboratory, the patient undergoes chemotherapy to kill the remaining blood-forming cells so that these cells don't compete with the transplanted cells and continue to produce the non-functional hemoglobin protein and allowing the disease to progress. The first gene therapy for sickle cell disease is a treatment known as Lyfgenia. In Lyfgenia, it adds a functional copy of the hemoglobin gene to the patient's cells, and this will allow the cells then to produce the working functional form of hemoglobin. The second gene therapy treatment for sickle cell disease is known as Casgevy. Casgevy is a situation where the DNA is edited in a way that allows the cells to produce a fetal form of hemoglobin that is normally turned off by the time an infant is about 1 year old. This fetal hemoglobin accumulates in the red blood cells and carries oxygen throughout the body. With the approval of these two gene therapy treatments for sickle cell disease, we now need to determine whether or not the insurance companies will pay for these procedures. Insurance uses a process called risk pooling to provide the funds for clients who have claims for covered services. The clients that purchase insurance are entering into a risk pool with other clients. However, not all individuals have the same need for covered services. Using health insurance as an example, the risk pool includes individual subscribers and family subscribers. Healthy individuals are pooled with unhealthy individuals. Actuaries use statistical models to estimate the financial risk in order to determine the insurance premiums for the health plan. The insurance company needs everyone in the risk pool to submit their premiums so that they can cover the expenses of claims. For the insurance to work, the amount of premiums collected must be greater than the amount of claims paid. In an October 2023 op-ed published in Bloomberg, MIT economist Andrew Lo provided an example of a typical health plan with 1,000 people, which will have an annual budget of about $6 million. Coverage of a single patient receiving gene therapy for sickle cell disease may use half of the available funds needed to provide insurance coverage for the entire 1,000 subscribers. In order to determine the cost effectiveness of a medical treatment, the insurance companies have to look at the costs associated with the illness. U.S. patients with sickle cell disease pay almost 4 times the out-of-pocket expenses over their lifetime compared to people without the disease. Insurance companies will pay out an average of $1.7 million for medical treatment over a patient's lifetime, which can be 9 times the average cost of patients without sickle cell disease. In addition, some estimates show patients with sickle cell disease lose around $700,000 in income because of their inability to work. The reductions in future medical expenses must then be compared with the immediate cost of treatment. The CRISPR treatment Casgevy will initially cost $2.2 million, while the gene addition treatment Lyfgenia will cost $3.1 million. Insurance companies must now determine if these treatments are cost-effective to provide coverage. These life-changing treatments can eliminate chronic pain experienced by patients with sickle cell and reduce the risk of other serious health problems, including death. Patients with sickle cell disease are expected to only have about 30 healthy years, compared with 64 healthy years for the average American. To help make the cost more affordable, both companies are considering outcome-based contracts that would link reimbursement for this treatment to how well the patients do. However, some insurance providers, such as Medicare, are not able to pursue this option at this point. So with this background information, we can now consider the ethical dilemma in science. Should insurance companies pay for gene therapy to treat sickle cell disease? In order to answer that question, we need to consider, should approval for expensive treatments consider improvements to the quality of life for the patient in addition to reductions in medical expenses? How do we ensure access to this treatment for patients with limited medical insurance, such as self-funded plans or state Medicare programs with small budgets? And then finally, there are roughly 100,000 patients with sickle cell disease in the United States, but there are an estimated 300,000 babies born with sickle cell disease in sub-Saharan Africa each year, where access to advanced medical care is extremely limited. Do countries with advanced medicine, such as the United States, have an obligation to provide this treatment available to developing countries without the medical resources to provide this treatment? Thank you for engaging in this important conversation about the ethical impact of science and technology on society. In the description below is additional information and references. Please comment with your thoughts on this topic or suggestions for future topics. If you enjoyed this video, please like it and subscribe for more conversations on the ethical impact of science and technology on society. Until next time, stay curious and be informed.